Hello, thank you very much for the opportunity to present our project today. We plan to cure idiopathic pulmonary fibrosis with thyroid hormone mimetics. Our team is led by Naftali Kaminsky, who is a world-known expert in pulmonary fibrosis research. My name is Milica Vukmirovic. I am a scientist and business developer. We have an award-winning team here, over 250 publications in top general and top pulmonary journals, over 40 million in NIH grants. And we have track record of discovery and innovation. Idiopathic pulmonary fibrosis is a lethal progressive lung disease of unknown origin. 190,000 patients in US and 6 million worldwide are affected with this disease. Median survival is 3 years. There are 2 FDA-approved drugs on the market, but they have significant side effects and no obvious impact on survival. However, they made sale in 2015 for more than $600 million. So the need here is to have a safe drug with a positive effect on survival and quality of life for IPF patients. Our key finding is that thyroid hormone agonism reverses pulmonary fibrosis. So environmental stress, cigarette smoke, infections, genetic factors are known causes of injury of alveolar lung cells. They cause injury of mitochondria, and they lead to epithelial cell apoptosis, fibroblast activation, and fibrotic process in general. Our solution here is thyroid hormone. We are actually curing mitochondria, restoring mitochondrial homeostasis, and repairing injured epithelial cells, and that leads to fibrosis resolution. Now, our approach here is to repurpose the well-characterized thyroid hormone mimetic sobeterum, this time to target pathways related to human fibrosis. In 1998, sobeterum was synthesized and currently is off patent formulation. From 2005 to 2008, was licensed to Quatrix for treatment of hypercholesterolemia. In 2010, Neurovia acquired and started development of X-linked adrenoleukodystrophy. In 2017, Neurovia completed phase 1 safety studies, and now it's recruiting adults and children for X-linked adrenoleukodystrophy treatment. They secured investors such as Novartis Venture Fund. So what this tells us is that we have a safe molecule to go into treatment with humans. Sobeterum resolves established lung fibrosis. As we can see here, this is the fibrotic lung upon the bleomycin treatment, and this is upon the treatment of sobeterum. Fibrosis is resolved. Sobeterum restores mitochondrial homeostasis. When we performed mitochondrial stress test, we saw that, bleomycin injured cells have low functional mitochondria, and when we added sobeterum, that this function restored to the level of normal. Now, patent for sobeterum was filed last year, and this year in June we'll obtain international protection. Our plan to develop sobeterum is to perform rat GLP studies to explore oral and local delivery, and to perform real-world dog efficacy studies. We are also engaging regulatory consultants, and we are negotiating with Neurovia to have option to have right to reference their data. By year 2, we hope to be ready for IND application, and then to go into the phase 1 studies in humans. Now, a little bit about our dog model. So these cute, these cute white terriers, they develop spontaneously fibrosis, as we can see on immunohistology and also on CT scan, honeycombing pattern characteristic of lung fibrosis. And they die, similar to IPF patients. This is our design, experimental design, how we would like to test sobeterum in these drugs. And we actually think that if we have successful efficacy studies in these dogs, it will be very helpful for us to have successful IND submission. Now, potential for further development of thyroid hormone mimetics to treat fibrosis doesn't have to be limited to lung fibrosis. We can open it up to liver, renal, kidney, cardiac fibrosis, because fibrosis is a terminal stage in most of the chronic diseases. So with this, we can help millions of people, and we can open market billions. And thank you very much for your attention.
First, my apologies that Milica obviously was presenting. Questions from the audience? Question here.
Hello, how does your dog animal model replicate the idiopathic nature of the disease?
Well, we will say it replicates it very well in sense of what we see on histology. It's very similar to what we see in IPF patients and also on CT scans. And they spontaneously actually develop fibrosis, which is really important. So we're not going with bleomycin.
Other questions? Judges? No? Do you have other molecules in the pipeline beyond repurposing an older one?
We are thinking about, yes.
Good. Well, thank you very much.
Yes, thank you.